everyone. Here we're going to start to focus on the electrical signals within the nervous system. Just to review a little bit, as you guys have learned already from the muscle tissue chapter, electrically excitable cells, such as the muscle cells and the neurons, they're going to operate through this ion movement across the plasma membrane. Remember when we talked about muscles, we were talking about the calcium ions. And as you will see, many of those principles that you studied for the skeletal muscle fibers, they're also going to be able to apply here to neurons. And with regards to the electrical signals, they're going to be produced by the nervous system, and they're going to be called action potentials. Therefore, electrical signals is the same thing as action potentials. And our ability to perceive our environment, to be aware of our environment, perform complex mental activities, and also to respond to the stimulus depends on these action potentials. So, for example, the brain will interpret an action potential that's received from the sensory cells as vision, hearing, touch. Remember when we were talking about the difference in the water temperature? This is all perception. And then this will go to the brain. So there's going to be a complex mental activity such as a conscious thought or a memory and emotions, or an action to remove your hand, for example. These are all going to result from action potentials. In addition to secretion from glands, these are all going to occur in response to action potentials that will be generated within these neurons or these muscles or these glands, depending on what the effector organ will be. Now, there's a basic knowledge of electrical properties of cells is going to be necessary for us to understand many of these normal functions and also the pathologies of the body. And the membrane potential is going to be defined as this measure of electrical properties of the plasma membrane, and it's going to be due to two major characteristics, which are the ionic concentration differences across the plasma membrane and also the permeability characteristic of the plasma membrane. Therefore, for us to be able to understand all this, I think it's a good idea for us to review a little bit of how movement occurs through the plasma membrane. So let's cover those in the next few slides. I know that on these next few slides, this content might be content that you guys have covered before in cell biology, for example, but I do think it's important to review, even if you have seen it before, because it will be helpful to understand when an action potential will occur. For example, to be able to transmit the signal within the neurons over on to the effectors. So here we're going to cover the transport across the cell membrane, and one of the great wonders of the cell membrane is its ability to be able to regulate the concentration of substances that are inside of the cell. And these substances will include ions such as calcium, sodium, potassium, and chloride, and things like nutrients, which will include, for example, sugars, fatty acids, amino acids, and also some of the waste products, which we can say carbon dioxide must be able to leave the cell. So it will be able to control what stays inside and what stays outside. We do have this membrane lipid bilayer, which is the structure that will provide the first level of control. The phospholipids are these structures over here, and they're going to be tightly packed together. And the membrane has what we call this hydrophobic interior. Hydrophobic just means that it doesn't really like water. So this will cause the membrane to actually be selectively permeable. So it will select what it wants to go inside. In the case of the cell membrane, only very small nonpolar materials can move through the lipid bilayer. Remember that the lipid tails of the membrane are nonpolar. So some examples of these are going to be other lipids, oxygen, carbon dioxide, gases, and alcohol. However, there are going to be water-soluble materials like glucose, amino acids, and electrolytes that need some assistance to cross the membrane because they're going to be repelled by the hydrophobic tails of this phospholipid bilayer. And all substances that move through the membrane, they do so by one of two general ways, either by passive transport or active transport. Passive transport needs no energy, so no ATP, and active transport does need energy. It does use ATP to be able to occur. Let's cover these methods in the next few slides. First, in order for us to understand how these substances will move passively across a cell membrane, it is necessary to be able to understand the concentration gradients and diffusion. Therefore, a concentration gradient is defined as the difference in the concentration of a substance across a space. So molecules, or ions in this case, they will spread or diffuse from where they are more concentrated to where they're going to be less concentrated until they actually reach an equilibrium. So there's going to be an equal distribution in both spaces. So when molecules move in this way, they're going to be said to move down their concentration gradient. So from an area that's more concentrated to an area that's less concentrated. And this is actually what defines diffusion. So diffusion is this movement of particles from one area of higher concentration to an area of lower concentration. And there are a couple of examples that might be helpful for you to understand this. So imagine that you're inside a closed bathroom, and if a bottle of perfume were sprayed, then the scent would spread all around the bathroom. So it would go from that first location where the scent was sprayed, and it would travel to all corners of the bathroom. So it would move down their concentration gradient by diffusion. Another example might be when you're putting sugar on your tea, for example. So you put a spoonful of sugar and you mix it up, and all the sugar gets dissolved. So eventually the sugar will diffuse throughout the tea until there's no concentration gradient remaining. And in both cases, if the room is warmer or the tea is hotter, the diffusion will occur even faster because the molecules are going to be bumping into each other and spreading out faster than at cooler temperatures. Therefore, having this internal body temperature around 98 degrees Fahrenheit will also help in the diffusion of the particles inside of the body. Now, diffusion can be divided into three. We have simple diffusion, facilitated diffusion, and osmosis, and we will cover those in the next few slides. So here we have the lipid bilayer. This part is the outside of the cell, the extracellular fluid, and in here is the inside, so the cytoplasm of the cell. Notice how you have all these molecules that are small and uncharged. This means that you're only going to have small substances like gases, for example, oxygen and carbon dioxide, that will be easily able to pass through this lipid bilayer and diffuse into the cell, as it will go from this more concentrated environment outside to a less concentrated environment inside. And because they are small, they move right in, and you can see at the end that we have the same quantity, the same amount of these molecules outside of the cell and inside of the cell. So this is what we call a simple diffusion. And as you can imagine, it does not require energy for this process, as it can easily move from one side of the cell to the other down the concentration gradient. Next, we have what we call the facilitated diffusion. Facilitated means that something will help out in this process. So what's going to help out are these proteins, which are either a channel protein or a carrier protein. The channel proteins, they're going to be less selective than the carrier proteins, meaning that they care less about what's going through them. So they're less selective about what's actually passing through them than the carrier proteins. But again, what will be passing through them will be moving from a more concentrated area to a less concentrated area down their concentration gradient. An example of this would be the sodium ions. So even though these ions are going to be highly concentrated outside of the cell, these electrolytes, they're going to be charged and they cannot pass through the nonpolar lipid bilayer of the membrane. Therefore, their diffusion is going to be facilitated by these membrane proteins that form a sodium channel, which can also be called pores, so that the sodium ions can move down their concentration gradient from outside the cell to inside of the cell. And there are many other solutes that must undergo this facilitated diffusion to be able to move into the cell, such as amino acids, or to move out of the cell, such as waste products. Now, because facilitated diffusion is going to be a passive process, it also does not require energy expenditure by the cell. So there's no ATP involved in this process. And water can also move freely across the cell membrane of all cells, either through the protein channels or just by slipping between the lipid tails of the membrane itself. And therefore, osmosis is the diffusion of water through this semi-permeable membrane. Therefore, we can see over here on this example, we have a beaker, and inside the beaker we have this semi-permeable membrane that's representing the cell membrane. And notice how the volume is the same on this side, but you do have more solute on the right compared to the left. So what you want to happen is you want to distribute the water according to the concentration of the solute. Therefore, you want more water to go to the right side. And this is exactly what's happening here. So at the end, you will have the same concentration of solute on both sides. Therefore, you need more water on the right side compared to the left side because you have more solutes on the right side. Therefore, this is what we call osmosis. So basically, it's the water diffusion through this semi-permeable membrane. Now, movement of the water molecules or osmosis is not itself regulated by some cells. So it's going to be important that these cells are exposed to an environment in which the concentration of solutes outside of the cell, therefore in the extracellular fluid, is equal to the concentration of solutes inside of the cell, therefore in the cytoplasm. Now, two solutions that have the same concentration of solutes are said to be isotonic. Therefore, they have equal tension, meaning that the concentration inside of the cell is the same as the concentration outside of the cell. Now, of course, when it is isotonic, there's no osmosis occurring, right? Because they are in equilibrium. Therefore, osmosis is only going to occur when there is going to be an imbalance of solutes outside of a cell versus inside of a cell. Therefore, we can say that a solution that will have a higher concentration of solutes than another solution is said to be hypertonic. And in this case, the water molecules, they will tend to diffuse into the hypertonic solution because you have more solutes on the outside. Therefore, the water will be moving to the outside to be able to form the equilibrium. And you can see when this happens, the cells don't really like because they become sort of dehydrated, right? Because there's less water inside of the cell. Now, in contrast, a solution that has a lower concentration of solutes than another solution, then it is said to be hypotonic. And therefore, the water molecules, they will tend to diffuse out of the hypotonic solution. Therefore, in this example here, the cells are in a hypotonic solution. So this means that the water will go inside of the cells. And if you have too much water, the cells will actually start to swell up. And with this, there's a risk of eventually these cells actually bursting. So it's a critical aspect of homeostasis in living things to create an internal environment in which all of the body cells are actually in an isotonic solution. And there's various organ systems, particularly the kidneys, that work to maintain this homeostasis, to make sure that everything is balanced out. So for all the transport methods that we described previously, the cell expends no energy, so there's no ATP that's required. However, during active transport, ATP is going to be required to move a substance across a membrane, and often this will occur with the help of protein carriers, and it's usually going to be against its concentration gradient, because it makes sense. If it was down the concentration gradient, then you would not need something to force a protein or a molecule to move, right? Because it will do through the other methods that we saw. But since you're forcing it to go against the concentration gradient, you do need to have something that will actively transport it, and therefore it requires energy. Now, there are three methods of active transport. We have sodium-potassium pump, we have endocytosis and exocytosis. So like I said, the active transport will definitely need ATP. It will be helped by a carrier protein. In this case, it's the sodium-potassium pump, which can also be called the sodium-potassium ATPase, and it's going to usually occur against the concentration gradient. Now, it's important to realize that we do have more sodium ions outside of the cell and more potassium ions inside of the cell. Therefore, the sodium-potassium pump, it will transport sodium out of the cell while moving potassium into the cell. So you can see how it's going against the concentration gradient because it's moving the sodium out of the cell where there's more sodium already. So it's not something that the sodium would like to do, right? So the sodium-potassium pump is actually going to be an important ion pump that's going to be found in membranes of many different types of cells. And these pumps are going to be particularly abundant in the nerve cells, which are going to be constantly pumping out sodium ions and pulling in potassium ions to actually maintain an electrical gradient across their cell membrane. So what is an electrical gradient? Well, it's going to be the difference in this electrical charge across a space, across the cell membrane. In the case of the nerve cells, for example, the electrical gradient is going to exist between the inside and the outside of the cell, with the inside being negatively charged, so at around minus 70 millivolts relative to the outside. So the negative electrical gradient is going to be maintained because each sodium-potassium pump will move 3 ions of sodium out of the cell and 2 ions of potassium into the cell for each ATP molecule that's going to be used. And this process is so important for the nerve cells that it accounts for the majority of their ATP usage. Now, other forms of active transport that do not involve membrane carriers are endocytosis, which actually means bringing into the cell. It's going to be the process of a cell that's going to be ingesting the material by actually enveloping it in a portion of its cell membrane, as we can see over here. And then it will pinch off that portion of the membrane. And once pinched off, that portion of the membrane and its contents, they will become an independent intracellular vesicule. And a vesicule is a membranous sac or a very spherical and hollow organelle that's going to be bounded by a lipid bilayer membrane as well, because remember, it pinched off from this lipid bilayer. Therefore, endocytosis often brings the material into the cell that must be broken down or digested. And phagocytosis is going to be an example of endocytosis of large particles, where in this process, the cell will be eating up these large particles that it engulfed. And many immune cells actually engage in phagocytosis of invading pathogens as they're trying to get rid of them. In contrast, we have pinocytosis, which actually means that the cell is drinking, so it will be bringing in fluid that will contain dissolved substances into the cell through these membrane vesicles. Now, these two methods, phagocytosis and pinocytosis, they're going to take in large portions of this extracellular material in a very non-selective way. But there are going to be cells that can regulate the endocytosis of specific substances via what we call a receptor-mediated endocytosis. So these receptor-mediated endocytosis, like the name says, they're going to be important because they will do endocytosis by a portion of the cell membrane that will contain many receptors that are actually going to be specific for a certain substance. And this is how they're able to regulate what actually gets endocytosed into the cell. Now, iron is going to be a good example of this, as it is a required component of hemoglobin, and therefore it's going to be endocytosed by red blood cells in this way. So iron is going to be bound to a specific protein that's called transferrin in the blood, and there are going to be specific transferrin receptors on the red blood cell surfaces that will bind this iron transferrin molecules, and therefore the cell will be able to endocytose the receptor ligand complex, as we can see over here on this image. Next, we have exocytosis, which is much like endocytosis, but it's actually a reverse process. So the material now, instead of staying inside of the cell, it actually wants to go outside of the cell. So the material that's going to be destined to be exported out of the cell, it's going to be packaged into a vesicle inside of the cell, and the membrane of the vesicle will then fuse with the cell membrane, and therefore the contents are then going to be released into the extracellular space. With regards to ionic concentration differences, we can definitely say that cells, they do have different concentration of ions in the cytoplasm when compared with the extracellular fluid around the cell. And it is this difference in ion concentration that will electrically excite the cell and use it to carry out their function. So as we know, there's going to be a higher concentration of sodium ions and chloride ions outside of the cell than inside of the cell, while there's going to be a higher concentration of potassium ions inside of the cell when compared to the concentration of potassium outside of the cell. Now recall that this distribution of ions is called a concentration gradient. Therefore, for sodium, there's going to be this steep concentration gradient from the outside of the cell to the inside of the cell, meaning that sodium wants to go into the cell. For potassium, the concentration gradient is the opposite of the concentration gradient for sodium. Therefore, for potassium, there's going to be a steep concentration gradient from the inside of the cell to the outside of the cell, meaning that the potassium wants to leave the cell because there are more potassium inside of the cell. And in addition to this high concentration of potassium in the cytoplasm, there's also a high concentration of negatively charged molecules such as proteins and other molecules that contain phosphate. So this makes the outside of the cell more positive and the inside of the cell more negative. That's why we say that inside of the cell there is a negative charge of around minus 70 millivolts. As we have seen, these differences in the cytoplasmic and the extracellular concentration of ions, they result primarily from this sodium-potassium pump and also from the permeability characteristics of the plasma membrane. So neurons are going to expend ATP to be able to maintain this uneven distribution of ions across the plasma membrane. Therefore, neurons, they expend ATP to maintain this uneven distribution of ions across the plasma membrane. And all along the neuron axon, the sodium-potassium pumps, they actively pump potassium against its concentration gradient into the cell, while at the same time they're going to be pumping sodium ions against its concentration gradient as well, but now out of the cell. And by transporting potassium into the cytoplasm, The sodium-potassium pumps, they're able to maintain this high concentration of potassium in the cytoplasm. And simultaneously, they're also going to be able to maintain the higher concentration of extracellular sodium ions. Remember that 3 sodium ions are going to be transported out of the cell, and 2 potassium ions are going to be transported into the cell for each ATP molecule that's going to be used. So now that we have reviewed all this, let's talk about how the plasma membrane of the neurons are going to be permeable to the passage of ions. So basically, they're going to go through what we call ion channels, and we have 2 different types of ion channels. We have what we call the leak channels, and we have what we call the gated channels. So in the next few slides, we will be talking about these 2 different types of channels. So the first type of channel that we have is what we call a leak channel. And like the name says, leak means that it will always be open, so it doesn't really control the ions that are coming in or going out. Therefore, we can say that they don't really have a gate controlling what's going in and what's going out. So there's not really an actual event that will open up the channel. Instead, it basically has this intrinsic rate of switching between the opening and the closing states. Now, these channels, they're going to be responsible for the permeability of the plasma membrane to ions when the plasma membrane is not stimulated, or in other words, when the plasma membrane is at rest. So there's no action potential occurring. So each ion channel is going to be specific for one type of ion, although the specificity is not very absolute. So they do allow other types of ions to go in, even though it's not specific for a specific ion. And the number of each type of leak ion channel in the plasma membrane will actually determine the permeability characteristic of the resting plasma membrane to different types of ions, meaning that the plasma membrane is more permeable, for example, to potassium ions and chloride ions, and it's much less permeable to sodium ions because the membrane has many more potassium and chloride leak ion channels than sodium leak ion channels. Therefore, there's more potassium and chloride going through the membrane than there is sodium ions coming into the cell through these leak ion channels. Now, the gated ion channels, like the name says, they will have a gate, and this gate will always be closed until it's opened by a specific signal. And by opening and closing, these channels can actually change the permeability of the plasma membrane. And there are 3 different types of gated ion channels. We have the ligand-gated ion channels, the voltage-gated ion channels, and other gated ion channels, which we will cover in the next few slides. So ligand-gated ion channels, like the name says, will have what we call a ligand, and a ligand can be either a neurotransmitter or a hormone. And these ligands, they will bind to a specific location, which is called the receptor site. So each ligand will have a specific receptor site where they will bind to. So in this case over here, we have a neurotransmitter, which is acetylcholine. Acetylcholine will bind to this specific receptor site that's specific for this acetylcholine receptor. And once it binds, it will open up these channels and allow, in this case, sodium to enter. So sodium now is outside of the cell because the channel is closed. Once the neurotransmitter binds to the receptor site right over there, it will open the channel, as we can see over here, and it will allow, therefore, mainly sodium, but we also have some calcium that's able to come into the cell. And because the channel is open, then you might also have potassium that's coming out of the cell, as we can see right over here. Now, these types of channels, they exist for sodium ions, for potassium ions, for calcium ions, and for chloride ions. And these channels, they're going to be common in cells of the nervous and the muscle tissues, as well as in glands, which makes sense, right? Because the neuron will be able to synapse with another neuron, with a muscle, or with a gland. Therefore, these channels have to be present in these three different types of structures. Now we have the voltage-gated ion channels, and these channels, they're going to respond to the changes in the electrical properties of the membrane in which it is going to be present. So normally the inner portion of the membrane is at a negative voltage. Remember we said, for example, for neurons is at minus 70 millivolts. So when that voltage becomes less negative, then the channels will begin to allow ions to cross the membrane. So actually here we can see that this voltage is at minus 70 inside of the cell. Therefore, this channel is going to be closed right over here. Once the charge of this membrane becomes less negative, so now it's dropping to minus 50, the channel opens and allows these ions to exit the cell. And they do so following the concentration gradient. So there's more of these ions on this side of the cell, so inside of the cell. And now, once the channel opens, it allows these ions to go to the outside of the cell. With regards to the other channels that are gated, we have two examples here. The first one is what we call a mechanically gated channel, and this has to do with actually a physical distortion of the channel. So once you have, for example, a touch or a pressure, you're physically distorting that channel. Therefore, the channel will open. So in this example over here, see how we have the channel closed? We will have way more sodium on the outside of the cell and more potassium on the inside of the cell. But once this channel is stimulated and there's a physical distortion of the channel or of the membrane, then the channel opens and it allows potassium to go out down the concentration gradient and sodium to come in down the concentration gradient. And that's how the ions will move inside of the cell. Now the same thing will happen with thermoreceptors. So once there's a change in the temperature, it will open up the channels and you will have one ion going in, one ion coming out. Now the electrical state of the cell membrane can have several variations, and these are all variations in the membrane potential. So a potential is going to be a distribution of charge across the cell membrane. And this, as we know, is measured in millivolts. And the standard is to compare the inside of the cell relative to the outside. So the membrane potential is going to be a value representing this charge on the intracellular side of the cell membrane. And this is based on the outside being 0. So in this example here, we have this electrode that's inserted into the membrane of the cell, and it's measuring the membrane potential on the inside of the cell. And this membrane potential is minus 70 millivolts, as is stated here. Now the concentration of the ions in the extracellular and intracellular fluids is going to be largely balanced, and it will have this net neutral charge. But however, there's going to be a slight difference in charge that occurs right at the membrane surface, both internally and externally. And it is this difference in this very limited region that has all the power in the neurons and also in the muscles to be able to generate this electrical signal, including action potentials. So this difference in the charge right at the membrane potential is what will create this electrical signal and then generate an action potential. Before these electric signals can be described, the resting state of the membrane must be explained, because or else it's going to be hard for you to understand. So when the cell is at rest, meaning there's no electrical current going through it, there's no action potential, and the ion channels are closed, of course, except for the leakage channels which randomly open, then the ions are going to be distributed across the membrane in a very predictable way. And we know how this is, right? We've been talking about this. So the concentration of sodium ions outside the cell is going to be 10 times greater than the concentration inside. and the concentration of potassium inside of the cell is going to be greater than outside. In addition, the cytosol will contain a very high concentration of anions, which are in the form of phosphate ions and also negatively charged proteins. And this is how we get the negative charge inside of the cell. So with the ions being distributed across the membrane at these concentrations, there's going to be a difference in charge, which is measured at minus 70 millivolts, which is going to be the value that's described at the resting membrane potential. So every time that we say that the neuron has a resting membrane potential, we will refer it to containing a charge that it's at minus 70 millivolts. Of course, this exact value is measured over here using this device, but minus 70 is most commonly used as a value, but it definitely can vary between plus 5 and minus 5 millivolts. So that would still be considered a resting membrane potential. So now that we understand resting membrane potential, we can move on and talk about action potential. When we're talking about resting membrane potential, we're actually describing the steady state of the cell, which is this very dynamic process that is going to be balanced by the ion leakage and the ion pumping channels. Now, without any outside influence, it will not change, because the leak channels, they allow ions to go in, ions to go out, and they sort of control that in a way that the membrane will always be at around minus 70 millivolts. Now, to get an electrical signal started, the membrane potential has to change. And this starts with a channel opening for sodium in the membrane. And because the concentration of sodium is very high outside of the cell, by actually a factor of 10, the ions will then rush into the cell, and this is actually driven largely by the concentration gradient. Now, because sodium is positively charged ion, it will then change the voltage inside of the cell compared to the outside. So the resting potential is going to be the state of the membrane at a voltage of minus 70 millivolts. So the sodium cations entering the cell, they will cause it to become less negative. And we can see over here as it goes from minus 70 to plus 30. And this is going to be known as depolarization, meaning that the membrane potential moved towards the zero. Now, the concentration gradient for sodium is so strong that it will actually continue to enter the cell even after the membrane potential has become zero, so that the voltage immediately around the pore begins to become positive. And the electrical gradient also plays a role, as the negative proteins below the membrane are going to attract the sodium ion. Therefore, the membrane potential will actually be able to reach plus 30 millivolts by the time that the sodium has entered the cell, as we can see here from this image. Now, as the membrane potential reaches this plus 30 millivolts, then other voltage-gated channels are going to be opening in the membrane. And these channels are going to be specific for the potassium ions. Therefore, a concentration gradient will act on the potassium ions as well. And as the potassium ions start to leave the cell, they're going to be taking a positive charge with it, and the membrane potential begins to move back towards its resting voltage. And this is going to be called repolarization, meaning that the membrane voltage moves back toward the minus 70 millivolts value that is of the resting membrane potential. So repolarization basically returns the membrane potential to minus 70 millivolt value that indicates the resting potential, but it actually overshoots the value, as you can see over here, because the potassium ions actually reach equilibrium when the membrane voltage is below minus 70 millivolts. So there's going to be a period of what we call hyperpolarization while the potassium channels are still open. And those potassium channels, they're going to slightly delay in closing. And this is what causes this hyperpolarization.